In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory.
Welcome to JAX Tech Talk with the technical information scientist of the Jackson Laboratory. My name is Corrigan Smothers, and my role on our technical information services team is to serve the scientific community with research education, solutions, and provide answers to all your technical inquiries. For anyone watching live, use the comment section of the video to share with us where you're viewing from and any questions that you may have. For today's talk, I'm pleased to be joined by Dr. Steven Rodriguez, Principal Scientist, Lab Head, Rare and Neurological Diseases at Sanofi, who will provide us with insight into how his group uses iPSC models to understand disease and how these insights can be leveraged for drug discovery purposes. Now, during his career, Steven used a variety of different mouse models and model systems, including mouse vivo platforms. And also, he used an iPSC technology, which is really a hot topic right now. Now, with that, and I don't want to steal his thunder of the introduction. Welcome, Steve. Tell us a bit about yourself and your work at Sanofi.
Thanks, Corrigan. Good to see you, and thanks for having me on your show. So I started my PhD in David Lynn's group at Cornell, working on neurodevelopment actually in mice. And we were interested in how neuronal connections are formed in three-dimensional space. However, none of the genetic models I worked on showed any neurodevelopmental phenotypes. And instead, one of the mouse models I was working on showed robust neurodegeneration as an adult. And this really opened up a new world for me. And I realized that we didn't know a lot about why neurons die prematurely in neurodegenerative diseases. So then I moved to Boston to do a postdoctoral fellowship in Mark Alber's group at Massachusetts General Hospital. And there I used the mouse olfactory neural circuit as a model to study neurodegenerative diseases as a postdoc. And during this time as a postdoc, I identified a link between the activation of specific innate immune pathway to neurodegenerative diseases. And this is what really started me thinking about how I could translate the findings that I found as a postdoc to develop therapies to treat patients with these devastating neurodegenerative diseases. And that's why I joined Sanofi, where I lead a research group that's working to identify new drug targets and therapies to treat these diseases. And I've been fortunate to work with some really good scientists, Disha Sood and Rachel Passaro, for example, on my team, who are really experts in iPSCs, and I've learned a lot from them over the past few years.
Nice. So tell me, how is your group using iPSCs, and how is your work leveraged by other departments at Sanofi in terms of drug discovery?
So I think iPSCs are one of several tools we use to model disease, but the reason why they're so powerful, the powerful tool, is because we can convert them to different cell types that we find that are in the brain that are thought to contribute to disease. For example, we use iPSCs to generate neurons, astrocytes, and microglial-like cells, and this is important because while we know that the clinical symptoms in most neurodegenerative diseases arise from the death of neurons, there's good evidence that these other cell types contribute to disease biology. And so with those cell types, once we've generated each of them, and we study them in isolation, but we start to also study them in combination, so co-culture. So you mix the cell types and try to replicate what's happening in a human brain. And these studies have allowed us to identify pathways that are dysregulated by mutations that cause neurodegenerative diseases. And then when we use this data to identify nodes in these dysregulated pathways that could correct that dysregulation. And hopefully those nodes would be sort of drug targets. And then we can circle back once we have a drug to sort of use these iPSC models to test our how efficacious our drugs are.
Awesome. So I'm curious. Why did you transition to iPSCs as a model for rare and neurological diseases. What did you use before the introduction of iPSCs? I mean, do you still use these in vitro models? And also when? I mean, what can you tell me on that?
Yeah, so I think I would call it more of an addition than a transition because we still use the tools that we used prior, but we add new ones as they become available. But we've turned to iPSCs for several reasons. I mentioned earlier about the ability to convert iPSCs into multiple different cell types that make up the brain. There are also rare mutations that cause neurodegenerative disease with a high penetrance. And we focus most of our work on studying these mutations across the multiple cell types to shed light on disease process. But another benefit of iPSCs is because most adult onset neurodegenerative diseases are actually sporadic in nature. So that means that there is no really clear genetic underpinning. These iPSCs, and we can generate these iPSCs from patients, they could potentially be used to model sporadic disease, which you really can't do with any other system. So this is kind of why we use iPSC, why we added that to our toolbox. But before iPSCs and currently, we really relied heavily on mouse models. And for our in vitro work, we use primary neurons and astrocytes from these animal models, or neuroblastoma cell lines, or a lot of the sort of workhorse cell lines that people use in the past, but they're limiting. And then, yeah.
That's interesting. So tell me, when working with iPSC lines, how do you choose your workhorse iPSC line? I mean, what kind of validation work or what kind of characterization are you looking for, or what's important to you?
Yeah, so that's a good question. So even though we focus on highly penetrant mutations, so the idea there is that the patient's background is not the primary influence for getting disease, we still see differences across lines. So it's really important for us to focus on the lines that are very well characterized so that when we do see changes, we know it's due to the mutations that drive the disease and not the result of cell line specific biology and not necessarily the disease biology. Does that make sense? Yeah.
Yeah, fantastic. So, and I know that our audience want to know this. Can you tell us more about, the phenotypes that you're interested in?
Yeah, so this is a complex question, but we study neurodegenerative diseases, so we really want to, the first question we want to know is, are there any clear degenerative phenotypes in a dish? But unfortunately, that hasn't been very consistent across models. But there are other cellular markers, for example, markers of organelles that you can measure differences in, markers of neuronal activity, and other markers that link back to disease biology, for example, protein accumulation, phosphorylation of specific marker proteins that might play a role in disease that we try to sort of learn from using the iPSC models.
So, how many different cell lines do you work with, to make sure that the phenotype is not specific to like a donor genetic background or the gender, or race for that matter, because when you're using an iPSC line, I mean, it's from like a specific donor. At what point do you take diversity into consideration here? I'm
Oh,
curious as to your answer on that.
Oh, these are good questions. They're very good questions. So, and it's difficult, and it's a difficult question because you have to have practical considerations when you're deciding the number of lines, for example. But I think it depends on initially the robustness of the biological phenotype you're studying. So robust phenotypes that can be replicated by that have been replicated by multiple labs and are known to occur in patients, then we don't worry so much about looking at many cell lines. However, for more nuanced phenotypes, you want to look in as many lines as possible. And so for us, we try to aim for, there's no set number, but we try to aim for at least 4 lines. But there are instances where you can only find lines from 2 patients, for example. So to give us confidence, we go back to other tools that we use and patient samples, importantly, to see what's consistent. And we tend to focus on those phenotypes that are consistent across the different model systems. And importantly, what really the patient data. So, postmortem brain tissues, cerebral spinal fluid data, whatever's really links back to the patient, and then that's that's where we put our emphasis on. And you mentioned diversity. I mean, and of gender and race, and That's clearly important. I talked about these very highly penetrant disease mutations, but there could be variants that are common to the underrepresented groups of females, or in my case, Latinos that we really don't understand, that could modify disease biology that we really don't currently understand. So I think this would help. It's important to understand, and this is where we hope to go in the future. So to look at, diversify our cell lines.
Yeah, yeah. And I know that you've touched upon this before, but how do you identify and go about narrowing down, like, say, a potential drug target?
No, that's I wish I had the true answer, but I can tell you how we. I'll tell you. So like I mentioned earlier, we take the data from all of our tools collectively, so we don't rely on any one piece of data. So, for example, we start off with patient genetics if we can, postmortem tissue samples, cerebrospinal fluid samples, and what we learn from modeling mutations that cause disease in iPSCs and mouse models. And we try to use all of this data to come to the best informed decision, because ultimately we want to be successful in bringing a therapeutic to patients, a successful therapy to patients.
Great, great, fantastic. So let's consider iPSC lines. And there are many out there and that one could choose to work with. So, if something is out of the ordinary, say in your experiments, you know, what are the most common causes that you can share with us?
Yeah, I mean, there could be many, but this shit has come across some practical ones, and that I think are pretty interesting. So one common issue that we found is when you're ingesting multiple iPSC lines from different sources. So different vendors have different methods of propagating their lines through different passaging methods or coding of their plates, and these different lines can have varying levels of spontaneous differentiation during propagation. So the unexpected but likely cause of the difference in these lines could be attributed to culturing conditions. And of course, passage number of cells. So how long one vendor has kept their cells proliferated, their cells versus another vendor, and spontaneous mutations can arise in the background that have nothing to do with the disease biology, but you can be fooled into thinking this contributes to what you're seeing. I mean, but contributes to what you're seeing, and you can be tricked. And so we've tried mitigating at least the most obvious thing is the culturing conditions, and this helps basically standardizing the culture conditions across all our lines when we bring them in.
Great, great. So, Stephen, this question just popped up, and I want to ask it before I forget. And in working for JAX, and I have to ask this. So, where do iPSCs fit in with mouse work? Is it just before doing like a in vivo preclinical study, or do you use them complementarily alongside animal studies?
I think you have to use them complementarily. I think, and we use them at all stages now. Ultimately, the fundamental question is, why are neurons in the brain degenerating prematurely? So, I come from a neurodevelopmental background, and clearly the cells in your environment influence what's happening to you. So we really need to understand what's happening in the brain first and foremost. And we're trying to recapitulate that with iPSCs by sort of bringing in multiple cell types into a dish. Taking the potential for those cells to become any cell type you can derive them into, particularly specifically for us, those that in the brain cell types, and asking how they influence each other in the dish, and then seeing how what you learn from the mouse, what you learn from the cells, and seeing what's consistent across the studies across those different models, and then ultimately, more importantly, how that links back to human disease. I think that's always the gold standard, the patients, and what the samples that they provide are telling us about the disease, and if those sort of markers or of disease come up on our assays.
Great, fantastic. And you've mentioned this, but I had a question that come in is like, what are your future goals for iPSCs? And, you know, are you planning to further develop your own iPSC platform?
So I mean, I can't tell you specifically what we're doing in-house, but you know, there are several things we want to do. I mean, broadly speaking, I mean, of course we want to expand the source of our cells. For a while, that was limiting. You know, you can only get them from certain sources. But now, for example, the Jackson lab now distributes iPSCs carrying mutations, different disease-causing mutations, that have been generated in a single well-characterized patient line. So this is the opposite of diversifying the line. But so there's two arms, you know, and what these cells allow you to do is ask, what are the nuanced differences that these mutations have on a single well-characterized line? So that could help us really potentially understand the nuances of the phenotypes from these different mutations, because you're in the same genetic background, and then complement that with increasing the number of the diversity of patient cell lines we get from patients, and then asking, you know, and then looking at them together. The other thing is that, you know, of course, we want to, you know, the more cell types we can get from the brain, the better. The better we can recapitulate that. You know, everything we do can be, you know, some people do, you know, tricultures, but in spheroid form. You know, you know, it's not something we're looking at now, but, you know, just trying to really understand how cells communicate in a dish. And then lastly, as tools evolve, just to look at the phenotypes, you know, sort of bring those in. For example, you know, we use single cell sequencing. And so now we could try to understand how for example, cells in our triculture are responding to each other, because we have the signature from the cells in isolation, and then we have the signature from the cells once they're together. And then we could potentially use AI tools, you know, cutting edge AI tools, to really extract the meaning of that data, and how it links back to human patients. So that's, I think, where you know, it's expanding the sort of diversity of the cell lines, focusing on the genetics in a specific cell line, and then bringing that together with novel tools to really extract the best data you can from those tools.
Fantastic. And Steve, I want to get to some questions from our audience that have been submitted here. And one of the questions that's asked is that, what kind of controls are you using when running iPSC experiments? Which is probably like a curious one that's out there.
No. I think ultimately, I mean, you know, I told you we focused on mutations that are known with high penetrance to cause the disease. So, and there's differences in different patient lines themselves for what, you know, either the culturing conditions or the patients themselves. So the ideal controls are isogenic controls, where you sort of take that patient line, correct the mutation, and then use that as your control system. In an ideal world, you could have hundreds of patients and hundreds of, you know, normal patients, but that's not, I think that's much more challenging.
Right. And does that approach, you know, help to, I guess, control for like off-target effects? You
Yeah,
know,
that's a good point.
Here. Yeah.
I think so that brings up an important point, I think, because when you make these isogenic controls, the modern technique now is CRISPR-Cas9, so genome editing, and those techniques themselves can cause potentially cause some genetic mutations that are hard to detect. So I know, for example, with the Jackson lines, they're using revertant mutation. So basically generating the cell line with the mutation and then back correcting it. So that way, any genetic modifications that occur during the process, you're controlling for in the best way possible. And I think that's a very powerful sort of control line to have. So both isogenic from the patient lines, but also this back revertant, which I think is important.
Awesome. And for those in the audience, please keep your questions coming in. And we have a question here. Let me see. I don't know. Feel free to throw this one back to me, Steve, if you want to. What approaches do you use for genetic manipulation in iPSC-derived neurons?
I think now, and the modern technique is you know, editing using CRISPR-Cas9, because it's efficient. It's you know, we're learning a lot more about off-target effects. these back corrected lines now help us avoid some of those mutations. So I think that's the primary genetic manipulation for generating for genetically manipulating iPSC lines, whether you're taking it from a patient and making an isogenic line or taking a standard line and making a mutation out of that line. So yeah.
Awesome. This is fantastic. And I see that those are the questions that we've had for right now. And to my audience out there, that's all the time that we have for this week. And I'd like to say thanks to Steve and also everyone for participating in this GTT. If you have additional questions that you didn't ask, you didn't pose in our chat, please email us at micetech@jac.org. That's MICETECH@jac.org. Also, please go to our website and check out our catalog of iPSCs, and that's at jac.org/iPSC. And don't forget to follow JAC Tech Talk on LinkedIn so that you're notified every time that we've got a new episode that's coming out. And also, or additionally, subscribe to our playlist on YouTube. So this is Corrigan Smothers and Steve Rodriguez signing off. Stay healthy, stay safe, and stay excited about research.
Thank you, Corrigan. Thank you, everyone.
Thank you, Steve. Thank you.